For the ones who are not familiar, in the early Drosophila embryo, this is as early as you can, just after egg laying, you'll see deposition or enrichment of certain mRNAs and proteins at the very posterior tip, which together is referred to as germplasm. It's just a part of the cytoplasm at the posterior end, which is enriched for a certain set of mRNAs and proteins. And then it at nuclear cycle 9, because there's a syncytial division at nuclear cycle 9, you have few nuclei that come in contact with this part of the germplasm, and they sort of bud out, externally bud out, and they undergo around 2 to 3 divisions, giving rise to all the set of cells, pole cells. And these are the first cells to sort of cellularize, because even the blastoderm hasn't cellularized yet. So in some sense, the pole cells are the first cellularized cells to arise in the case of Drosophila. All right. So much of today's talk will be about how does this stuff that is highlighted here, which is a cocktail of several mRNA and protein, get shared between these. And before that, you might ask, why is that relevant? Let's just play this and see what happens to them at some point in once they're formed. Okay. They go to the gut, they exit the gut, they go to the gut because of gastrulation, they exit the gut, and they form two bilateral clusters, and they move, and they go and join a bunch of cells called the somatic gonadal cells at the gonad, and they form these two gonads. The video is actually smooth, it's just the presentation. So, and then earlier work showed that the amount you inherit in that germplasm, how much you inherit kind of dictates your chances of making it to the gonad successfully. So what did they do? They took the bunch of cells, and if you look from the anterior side from the top, they made two concentric groups, the central versus the peripheral ones. I made two different groups, and then notice that if you look for different germplasm mRNAs, there is more of it in the central ones, and there is less of that in the peripheral ones. And this also made a big difference to their eventual chances of making it to the gonad. And so if overall for the whole population, it's around 66% chance of in the melanogaster making it to the gonad, whereas in the central cells had a higher than average, which is 82%, and the peripheral ones had around 46%. So this is by taking the collection of cells, which are around 40 cells in melanogaster, and splitting them into two concentric groups, and just basically asking, what are their odds of making it to the gonad? And they saw that what makes them distinct is that how much they inherit is different, and the chances of making it to the gonad also changes. So in a sense, they established that the amount of germplasm kind of is proportional or influences survival. Survival means that how well can you migrate and reach the gonad. All right. I need to also mention the fact, which I will try to connect in the talk, which is that this germplasm is not like a continuous fluid, but rather these are very granular in nature. They are like, if you do an in situ and do an high resolution imaging, you'll see that these are like these, you can call them condensates, or you can call them granules, aggregates, which basically have a bunch of mRNAs and proteins forming this discrete quantize. It's like, in other words, one way to look at it is this cytoplasm is like, in a way, it's just a packet of mRNAs and proteins that are sort of distributed nicely. And not just that, people have also, this is a more recent electron micrograph showing that these are quite big structures, which you can see them under an electron microscope. And in the 60s, starting in the 60s, Tony Mormol actually did this in many different Drosophila, because he took them because it's EM, so it didn't have to have all the tools or fancy tools. You just needed to slice them and just basically look at the granules. And when he looked at it, and he published a series of papers on the nature of this, these germ granules or polar granules. Um, they differed in sizes across species, and they also changed. Their morphology kind of changed during the course of the development. So they fused, they basically grew bigger, and they were different in, like I said, Drosophila immigrants compared to Drosophila melanogaster, and they were bigger in immigrants. And so these things were characterized in a series of papers in the 60s. And not much after that, nobody sort of like cared so much about it. Not that I have done anything more, but the point is, it's a series of nice papers on this, and big gap after that. Okay, so I have two questions. One is how is the germplasm partition between the pulses? Why do I care? Because we know that how much you inherit kind of influences your chances of making it to the gonad. So it's kind of irrelevant. It's just a pointless. It's not a pointless bookkeeping. It's actually important. I know it's kind of tells the germ cells ability to migrate successfully. Without getting into the nitty-gritty details as to how exactly does it influence his ability to go to the migrate properly and so on, which has been looked at to some extent. And the second thing is, okay, now that I mentioned something that was slightly unrelated, which is the size of these granules, can this play any role at all in how things get distributed between the germ cells or the pole cells? I'll be interchangeably using it here, and for the time window in which I'm studying this process, they mean the same thing. Technically, I should be calling them primordial, PGCs. All right. So we knew some things before we went and basically made this process in greater detail. We already knew something from literature. Enough for us to sort of speculate about how this division of these pole buds giving rise to pole cells, how to model it. One, we say that there is a net amount of germplasm, which we showed, has to be finally shared between all the progenitors from the N0 number of initial buds that forms, because the buds have to undergo 2 to 3 divisions, given as the full set of cells. So even this total amount of G naught has to be distributed between them. Since we didn't know any better, we just assumed that the initial distribution between the buds is a multinomial distribution. I mean, when in complete doubt, you can assume that it is the P for each one of these buds is the same. And we assume that the subsequent divisions are subsequent sharing of the germplasm or the germplasm between the daughters is again like a binomial process. What I mean by binomial process here is that if there are n granules, each granule has PB probability of going to one of the daughters. So if I take 0.5, that's like a very symmetric division. And here comes one of the biggest assumptions, which is because we know that at some point in time, they don't keep on dividing. I mean, you guys can say that that's not the only reason, but they don't divide all the time. They at some point stop dividing. And we know that it's like cleavage divisions, and the stuff that they have inside keeps decreasing with time because they're dividing. So maybe there is a threshold below which you stop dividing. So we just assume that there might be a threshold, which we refer to as G dagger, below which the growth rate or like the division rate is 0. One big assumption.
So why did you assume that the number of division is controlled independently of GFP?
We didn't assume that.
Yeah.
It could be that, because the gastration starts right after and something is ending. It's one of the hypotheses. I guess the reason I'm presenting is because somehow eventually we gathered some more, evidence for it. But it is true that's not the only assumption. If you're in complete darkness, there are several reasons that might lead to 2 to 3 divisions. But this was one of the ones that.
This was assumed that if you don't have germplasm, you would not have any division. Or you would have much less. If you reduce the level of germplasm in the experiments, you will have less division.
It is true that there are other nuclei that continue to divide even though they don't inherit germplasm. I think this is a thing that we and other group I've discussed, which is that I think the cell cycle regulators in the two types are quite distinct. And then there are, I will mention this, there are during this division, sometimes you inherit so little that you actually stop being a germ cell and go back to being a blastodome cell. That such a process does happen. So in some sense, it's more intricate. I mean, definitely, it looks like they might be switching between this thing. So, and there's also an earlier work that kind of links at amount of this germplasm that gets taken to the nuclei, influencing whether they will divide or not. So the earlier, there are some reasons for us to sort of bias, to be biased towards this assumption than others. Okay. And I also made on top of that, just to simulate and to have some sort of sense, I made another assumption, which is on top of this, which is that, okay, given if there is a G dagger, below which there is no division rate is zero, maybe the dependence after that on the division rate or the growth rate, division rate is just linear. Why linear? Because if I have more exponents, more parameters to fit if I take non-linear. So let's start with a linear model. So this is like something that we assumed. These are the set of this thing. It's a very simple process. You can simulate it exactly, stochastically exactly because it's too simple. That's what we do. We do that, for example, in that model, you will see things like this. If I start with like 8, n0 is equal to 8 buds, there is a certain amount of G naught that gets split between the 8. The initial divisions are usually faster, naturally, because that's when they have the highest amount of stuff, and that's when the growth rate is the fastest. So they grow faster. And if the daughter, after the division, if there is any daughter which still has G greater than G dagger, it will continue to divide, and so on. And the ones basically, even after the second round of division, if they still have enough crossing G dagger, they will continue to divide. And so the divisions naturally become slower and slower with time, which is not shocking because just we have made the growth rate and it resembles what we see. It doesn't prove beyond doubt that this is what is happening, but we do at least get back what we see empirically, which is that there is a slowing down of the divisions with time. And people also observe that the first round of division it's synchronous, and later on they lose synchrony. But we think that the synchrony is just a consequence of the fact that the first round of division is faster. So the small variation, if you were to scale the variation by basically the rate of division, it's probably the same for all of them. It's just that doubling. So if you take the linear model, after every subsequent division takes twice as long because the amount of germplasm is becoming half, and we assumed a linear this thing. So naturally, the time window is now double. So you will notice asynchronous things more than what you do in the early division. So the early division does look more synchronous naturally because of this. I like to highlight another point. As far as if you think about it, the only thing the linear dependence on the rate dependence on the germplasm, the linear dependence, only thing it contributes to is basically when these division events happen. As far as germplasm sharing is concerned, if I took a model where it didn't care about, it just divided till basically as long as you were above G dagger, you divided and stopped when you went below G dagger, the division process is identical. Except that the only thing that comes from the linear dependence is the timing of things. So there I should see a division that is happening regularly at the same time, this thing, which we at least know is not what is happening. But as far as the division of the G naught between all the pole cells, doesn't matter. If you assume a threshold, you will basically, the process is very simple, which, um, which slide am I in? Okay. Okay, just to highlight, the process is very simple. You keep divide if you are, and if you end up with something more than G, if the G is still more than G dagger, you divide. That's essentially the whole thing. Now, it's just basically another way of showing the same thing that I showed you, which is that the subsequent divisions basically take longer and longer time. Here I also wanted to make one more point that if you start with a fewer buds, the system will just simply make them divide on average more number of times. So typically in Drosophila you see 2 to 3. Here just there are ones that have done 4, partly because I started with fewer birds than what typically Drosophila starts with. You just basically undergo one more round of division. All right. So there are a few things that we can easily get back from even this very simple model. One is, if I increase G naught, is there a way to increase G naught? The answer is yes. We can, for good approximation, changing the dosage of a one particular gene, the master gene that organizes the entire germplasm, which is Oscar, by just having, instead of having two copies, if you have one copy or three copies, you basically get, we know that you get fewer and more germplasm, no, sorry, pole cells. And if you count them empirically, this is what you see, which is that nicely you see around 50% decrease in one copy of Oscar and 50% increase in three copies of Oscar. Don't ask me what happens after three. That's for another day. So, but in the model, when you just basically say, when you keep the G dagger the same and just change the G naught, which I've taken the liberty to say that that's what is happening, you will see that in the model also just nicely reproduces the same thing, which is that the means just shift basically. It becomes half, it becomes half the amount and increases by 50% when you just change the G naught. Okay, this is just to reiterate the point, which is partitioning itself is agnostic to the rate of division. Anyway, now that we've made some this simple stochastic model and add some simple predictions, we wanted to basically go to the actual system in vivo and ask, are these assumptions justified? For which, this is where Shui comes in, and we realized that we need to be able to quantify the amount of germplasm very well. So we thought the better way to image the embryo is this way, which is upright, or en face, as some people call it. So, it's just, the hope is to generate videos that are, many videos of this, of course, with Z. I'm just doing a max. This ruins the video, but take a while. So now, what? Yeah. Thankfully, I quantified everything, and I'm not going to show anything based on this, because that would have been impossible under this circumstance. So this is where the first two summer students I showed you in the beginning came, because it's really painful to track these cells, and you need to sit and basically track these cells, and that's what that's what we do. And sometimes some of them, one of the daughters just stop being a germ cell because it has inherited so little, it just goes and becomes part of this blastoderm. So all of that happens. So obviously the best way to track is to start from the final state and just go back and just basically establish all of this. Once you do this, you get this lineage, and it does change. The number of lineages change because it's just basically a function of number of pole buds you start with. And on average, you do divide like 2 to 3 times. Okay, once you have that information, then the natural thing is, can we segment? I mean, get the 3D segmentation, which we can. Yeah, I mean, I think Vanessa knows what life cycle is. So we just. I feel your hand. So we get the 3D segments, 3D mass for these things, which is very necessary for us. And then once we have the lineage and the 3D segments, we can go back and basically obtain the amount of Vasa, which is our proxy for germplasm, which might have introduced some of that assumption. Often in the field we do use it, but then again, how good is it an approximation for every mRNA and protein that is there in the germplasm? I mean, but any one component would be as bad as this, or probably worse. So we just stick to Vasa, and we can basically get these things. And we have all the information about all the division events. But before that, let's go back and basically the original paper, which basically made concentric two groups of like central versus peripheral. So we can do the same thing. Just draw them radially two groups, roughly of the same size, and ask, is it true that the central, because it's also a sanity check for our data to see if we do see it. Of course, the central ones have more. and the peripheral ones have less. But you'll see that if you observe the whole process, the cells do mix a bit. So it's not, as an ensemble, you can make the statement the central ones have more. But if you see the distribution, it's quite varied. But it's okay. So that statement they made is still true because it holds for the group. But if you see the process, there is a bit of shuffling of their location and so on. It's not like the bud that is in the central will populate will give rise to all the central pulses. It's not like that. There is a lot of mixing. And it's not because how amount of germplasm you have is not simply because of volume. It's simply not a consequence of just having more volume or something. There is a scatter plot I can show you just to show between them. It's very weak correlation. Volume does play a role. Of course, the bigger the volume, the higher chances of the higher the amount you enclose. But that doesn't explain everything.
But is volume distributed like Vasa? Is it bigger in the center?
Good question. I can quickly just show you the correlation. I should probably show you a spatial information, but okay, I'm showing. Okay, now there you go. So if I take this, this is how the correlation between the fraction of the total volume and the fraction of the total Vasa looks like. It's not the strongest of correlations, but you can say that it is true that by and large, if you have more volume, you most likely have also more this thing. But what I can say is that I can't use volume as a proxy for Vasa. That's what I mean. It's better to just work with actual Vasa, total amount of Vasa itself in the volume. Okay. Question is this process finally, what does it do to the whole, in the beginning, we know that the pole buds take up a bunch of germplasm and they finally share it between themselves and finally give rise to basically all the pole cells. How does as a fraction of the total germplasm change and inherited by these things change with time? As in, like, if we take the initial state and the final state, how does it look like? This is the thing. Just to quantify, for the 4 samples at least, we did just compare the initial CV versus the final CV, which is the coefficient of variation, standard deviation by the mean. It's not necessarily something that decreases the variation. It actually slightly, if anything, it increases the coefficient of variation after the whole division. Okay, this is the biggest assumption. It is a bit of a leap to make this assumption that is the division rate proportional to G. Once I track all the division events and I ask how amount of stuff that is there and how long did it take before it basically went to divide, I basically get that information. So I basically have the rate, I basically have the amount of the Vasa, provided I have normalized and whatnot. It's like, all of that could have contributed to error, but finally the data looks like this. Important point is that there's a lot of points here. It's not very obvious, but when you do a locally weighted averaging, so if you do the lowest, that is, you do get the rough trend of what might be happening, which is basically this, which is very similar, not very similar, it's just basically the identical thing that I assumed. But of course, the process is very noisy because what you see is a lot of variation around this, underlying, function between the amount of germplasm, which is represented by Vasa, and our division. So I would say it's a weak yes. So it looks like it is proportional. Now the question is, are the divisions symmetric? So I can again do the same thing. Instead of showing all the, just as one group, I'm just showing the division events as a function of time as well. And because based on the amount the daughters inherit, I can get an F share, which is basically take one of the daughters' amount divided by the total sum of both the daughters. So if I get 0.5, that means that both the daughters are getting like equal amounts. And if I do that, you'll see that you can almost see from here itself that the asymmetries are greater in the beginning, but later on as the divisions go on, they become more and more symmetric. Just basically estimate the P using Bayesian methods. We get this, which is early, shown here by diamonds, is let's say something like 0.57, and late ones are much closer to 0.50, something like 0.52. And if you take the overall average, it's something around 0.55, which means at the early stages, it does get close to 0.6. And at the later stages, it gets very close to 0.5, being very symmetric. But it's not like dramatically asymmetric, but it is still asymmetric, especially in the beginning, which you could have seen, which you kind of can see it from the very beginning was. All right. So now we kind of get a rough understanding of what might be happening, how things are getting shared and so on. So we said, okay, so now when I started this, I assumed that the pole cell numbers would be wildly different across many species, many Drosophila species. So the only way to know was to basically stain them. Thankfully, we had some cross reacting Wasa antibodies. I used them and I stained them in a bunch of species and I counted them. And I counted them. Disappointingly, you'll realize that a lot of them, people who know flies, they know that a lot of them belong to Sophophora. It's not that different at all. If you just take the medians of this, it's not that different at all, except for a couple of species where we see it's actually lower, below 30. But by and large, it's variable, partly because my counting is not perfect, partly because it's actually variable. And it's not that distinctly different in all these species. But what we can also do is go and count the number of germ cells that make it to the gonad. So when you do that, you can then ask, by the way, this stage is even harder because I wanted to count at a stage where they have actually coalesced into the gonad, which is the hardest state to count because it's too dense. Counting it earlier the problem is not all of them who are made there is guaranteed to join the gonad because some of them will overshoot the gonad. So to be sure that they have become gonad, you have to wait. But that stage is also not the best time to be counting the cells. But anyway, given all those other things, you do see some sort of difference in the fraction of them that make it to the gonad. The ones who made fewer seem to have greater fraction of them making to the gonad. Okay? Now, making a big leap of argument here. So, and just to say that now, if you go back to the fact that Tony Moll's work on how these different species have different granule sizes, the one way to look at it is like, let's say if we were to take the initial scenario of the pole buds sucking up the germplasm next to them, and if I were to do just like a weird Warner tessellation of like what to highlight the germ granules that are closest to each of the nuclei, as opposed to this scenario, as opposed to this scenario, the amount of space occupied is the same. It's just that one is much smaller in area. So you would expect that because of the bigger granule sizes, you might expect greater variability in how much is inherited between them. It's just because of it's just bigger chunks, and you're sharing bigger chunks, and you're more likely to have greater variance. Question is, such a thing, can that contribute to division between daughters? Division of or partitioning of the germplasm between daughters. Can that make it more asymmetric? Because at least in a simple theory, we know from a simple model, we know that does play a role based on what is the like the p-value for the binomial process of splitting the granules between two daughters. So it does play a role in the final cell numbers, and I'm not showing it, but it also plays a role in the variability of the final amount of germplasm that is shared between the daughters. So it's not very hard to imagine why, when you have the same amount of stuff but packaged in fewer units, bigger packets, the process might induce greater variability in the sharing process. So we are asking, is that what is happening? Something of that sort is happening between species? Because we do know that their morphologies are different from the old electron microscopy images. Okay. How do we answer this question? what we know is, fortunately, is that the Oscar is the key germplasm organizer. And in the past, McDonald's group have actually expressed, for example, Virilis Oscar as well as the Drosophila immigrans Oscar in the Drosophila melanogaster. I mean, of course, they were asking slightly different questions, but at least in the immigrants paper, they show that the morphology of the germplasm does change because you have an Oscar from a different species that seems to have a different morphology being now expressed in melanogaster. So if the question is, can I use Oscar from other species expressed in melanogaster as a way to sort of tune, as a knob to basically change the morphology of the germplasm and hopefully see what happens. So that's what I did. I took a bunch of these pieces. I amplified the Oscar from there and just basically do 5C31 injection and just basically make these Drosophila lines, Drosophila melanogaster lines that express Oscar from these species. Why these species specifically? Just sometimes the answer is as simple as they were in the lab and I managed to basically clone it on time to send it for injection. Nothing profound, but I mean, I think we could sample more carefully, but I didn't. So the way I look at it, at least, is that you take Oscar, change this thing sort of as a knob to sort of control the granule sizes, and ask the big question: can we quantify, can we study the effect of this partitioning as a function of the granule sizes in some sense, the morphology, and how does it impact the sharing, and do we see greater variability or less variability in the final amount of germplasm the pole cells inherit if I were to study with these Oscar from different species in the melanogaster background? Because we can do the exact same pipeline because it's in the melanogaster now. All tools are available. All tools that are available for melanogaster are available. So that's one advantage. But it's not the same thing as getting the word studying in the native species. We know that. All right. To conclude, show that the rate of pole cell division seems to be a function of germplasm, linear, and loss of synchrony, which people talk about a lot after the first round of division, seems to be just a consequence of basically this rate of division being, if you were to say that the rate of division is a function of the amount of germplasm, then this is obvious. It comes from it very obviously. And the pole cell divisions are largely symmetric, at least in melanogaster, which might not be the case in other species, but it is largely symmetric. And larger granule size could make the divisions more asymmetric. That's at least the working hypothesis. And the asymmetric divisions could exaggerate the inequality in the partitioning and affecting the pole cell survival. So if you make it more fairer distribution between all of them, maybe then you improve the whole group has just a higher chances of making it to the gonad. As I said, in other species, there are a lot of things that are different because every other component of the germplasm is different and the embryo shape is different. And that a lot of things are different. So it's very unlikely that everything will nicely boil down to one or other thing, but we think this is a contributing factor. That's the hope. And expressing Oscar in melanogaster background is a way of basically just trying to understand the effect of just that on this whole process. So this is where we end. And I would thank all the members of the External Lab, people who fund me, and finally you guys for listening to me.